oxygen constantly. If they don't have oxygen for a matter of about 4 minutes, they'll start to die, like the brain and the heart itself and the kidneys. And so it's absolutely critical that we maintain this circulation. The heart really is composed of two sides, a right side and a left side. The right side pumps blood that is deoxygenated or less oxygenated to the lungs, to the pulmonary circuit, comes back to the left side, which then pumps it to the rest of the body, the systemic circuit. When we look at the heart, you recall that it has 4 chambers, 2 atria, which are the small chambers, and 2 ventricles. The blood comes back into the heart, into the atria, and then it goes through the atrioventricular valves into the ventricles. The atria contract and help fill up the ventricles with blood. Then the ventricles are the big pumps, and they pump the blood out. The right ventricle to the pulmonary circuit, and the left ventricle to the systemic circuit. We have a total of 4 heart valves, 2 atrioventricular valves. On the right, it's the tricuspid, and on the left, the atrioventricular valve is the bicuspid or mitral. The exiting valves from the ventricles are the semilunar valves. On the right side, it's the pulmonary semilunar between the right ventricle and the pulmonary trunk. And on the left side, it's the aortic semilunar between the left ventricle and the aorta. The left ventricle is thicker than the right ventricle, and that's because it has to pump against a higher resistance, higher pressure systemic circuit, as opposed to a lower resistance, lower pressure pulmonary circuit. Over time, though, the amount of blood that goes through the left ventricle and the right ventricle is equal. It's a closed circuit. When we look at the heart, we see that we have 3 major layers. The layers right by the chamber where the blood is flowing through is called the endocardium. This would be the deepest layer. The endocardium has an endothelium associated with it. Endocardium and endothelium are not the same. The endothelium is that simple squamous single layer of cells that lines the blood. And the endothelium is a subdivision of the endocardium. We'll also have generally some connective tissue in the endocardium, and sometimes even some smooth muscle, some Purkinje fibers, which are conduction fibers, modified nerves, if you will. And that's the endocardium. The biggest layer is the myocardium, and that, of course, is made up of cardiac muscle. We've looked at cardiac muscle, and we'll look at it again here, but we know that we have intercalated disc in the cardiac muscle, that it has branching, that it is striated, that it usually has 1 or at the most 2 nuclei. We can have some hormones released from the heart. The atrionatriuretic hormone is the most common, but we have some others there listed. The epicardium is the outermost layer, and that, of course, is going to be mostly connective tissue. The outer layer of the epicardium is the visceral layer of the pericardium. You may remember that we have a pericardial sac or pericardium around the heart that the heart sits in, and it has 2 layers, this membrane. The inner layer is the visceral pericardium, and that is the outer layer of the heart. So it's also the epicardium. It's the same thing. And then we also have the parietal layer of the pericardium. In most of our slides, I think all of them, the parietal pericardium is removed. It doesn't withstand the processing production of the slide. But we should probably be able to see the visceral pericardium, aka also known as the epicardium. We have in addition to these layers, we have the fibrous skeleton of the heart, kind of holds things together, connective tissue, made up of collagen fibers. For instance, the heart valves connect to this fibrous skeleton. This now shows you a slide from your book, and this is the endocardium and a little piece of the myocardium. Right here, we would have the blood in the chamber, and then this would be our endocardium. Lining right there, lining that chamber, is the endothelium. And you can see we have a single layer of flattened cells, so that's the endothelium. And then we can have other structures like some collagen fibers. These are Purkinje cells, or Purkinje fibers. Again, Purkinje fibers are a type of modified cell that is really good at conducting impulses. It helps move the electrical activity of the heart throughout the heart. And so those are Purkinje fibers, and that's shown right here. And then we get on into the cardiac muscle. So right about here, it would be the myocardium, and we're starting to see the cardiac muscle. This is H&E times 300. Here is a rather more extensive view of the endocardium. In our slides, we probably won't see all of these details. This happens to be one that catches a lot of details. But here we see in this orange line the endocardium. Here's the myocardium starting at the red line. And then here's our endothelium lining going up and down here, the flattened cells. Here's a fibrous layer with collagen fibers. Again, here's Purkinje fibers, and this is what they look like. You may find that. And then we have some more connective tissue here and some fat cells. So that's the endocardium. On the other side of the heart, we have the epicardium. That's from here. We see the laser pointer up to here. The epicardium, here's the myocardium down here, the cardiac muscle. The epicardium, you can see here, has a lot of fat and adipose. We also see some other collagen fibers up here. And here is the visceral layer of the pericardium, which is part of the epicardium. Notice the flattened cells. Here they're calling it a mesothelium, which is another name for it, where you have this layer. This happens to be a coronary artery. Some of the main arteries that carry blood to the heart itself are the coronary arteries, and this is an example. So the CA stands for coronary artery. Here's another artery here. Again, we have coming here, we have the heart valve, and it attaches into the fibrous skeleton, this connective tissue here. And here we have the endocardium, here, here's some myocardium, and here's the epicardium. This is a fairly low magnification, 120. Here's the heart valves, two atrioventricular valves, two semilunar valves. And in their interior, the valves have connective tissue. This looks kind of like dense irregular. And then we have an endothelium on top, on the surface of these these heart valves. This shows you the heart valve coming here and here, and again we have connective tissue inside of it, comes to the fibrous skeleton, and then we have an endothelium on the surface. This is a blown up view of that with the connective tissue in the interior and the endothelium on the surface. This is a picture from the book, very low magnification, only 15, magnified 15-fold, but we can see the valve here with the endothelium and the connective tissue coming to the fibrous skeleton here. In order to generate the heart rhythm, the heart beating, the heart, you may remember, is autorhythmic. You have the sinoatrial pacemaker, and it automatically regenerates action potentials, and then they spread first over the atria, then to the ventricles. And so the depolarization wave can spread from one heart muscle cell to the next through gap junctions that are part of the intercalated disc. And so you can have this wave. This is also going to be aided by the bundle of His, or the bundle branches, and the Purkinje fibers. So the movement of the depolarization wave, it goes from cell to cell in the muscles, but it is also helped by the bundle branches, also called the bundle of His, is how we pronounce that, and the Purkinje fibers. Although it's autorhythmic, the heart has its own rhythm. We can increase the heart rate using the sympathetic autonomic nervous system. This is coming from the cardiac center in the medulla oblongata. We can also reduce the heart rate using the parasympathetic system from the cardiac center in the medulla oblongata as well. So we have our general normal resting rhythm automatically controlled, and we can crank that up like for exercise, and then we can crank it back down after exercise. The rhythm would start here in the sinoatrial node and spread through the atria, pause briefly at the AV node, and then shoot down the bundle branches, the bundle of His, and around the Purkinje fibers, and cause this integrated contraction. This again is our endocardium, from about here to here. The blood in the chambers would be here, and then we have the endothelium lining that. These are bundle branches, the bundle of His, or bundle branches, modified cells to be very good at conducting. And then this is myocardium starting here. Here's our Purkinje cells. They have this very distinct look, and that's what they look like. Here's our endothelium, and here's our Purkinje fibers in the endocardium. Notice this is 400 magnification. Oftentimes these Purkinje fibers stain a little bluish gray, as we see here. These are Purkinje fibers. They kind of look like neuronal cells, like here. And then we have the cardiac muscle surrounding it and connective tissue. This is a nice view of Purkinje fibers. Notice they kind of look like neurons. This would be the pale staining nucleus and the nucleolus. Here we see another one. In this stain, we see some blood vessels. The red blood cells are stained brown in this stain. So this is a little capillary here. Here's another one. Here's another one. we're looking at the blood vessels, we have essentially the arteries, arterioles, mid-arterioles, then the capillaries, the venules, and the veins. The arteries carry blood away from the heart. In the systemic circuit, they're carrying oxygenated blood. In the pulmonary circuit, they're carrying deoxygenated blood. The veins carry blood back to the heart. In the systemic circuit, they're carrying deoxygenated blood. And in the pulmonary circuit, they're carrying oxygenated blood. We can divide these vessels into the largest of the arteries, which are elastic arteries or conducting arteries, and then the main largest number of arteries, which are the distributing or muscular arteries. And then we go down to little smaller arterioles, and eventually to capillaries, and then veins and veins. If we look at the total cross-sectional area, even though the aorta is much bigger than the other vessels, especially the capillaries, much bigger, we have so many capillaries that their total surface area is much larger than the aorta. So the surface area peaks at the capillaries because of their number, and then diminishes. The main velocity is in the arteries. It goes down to the capillaries and comes back up a bit to the venules and veins. We do have a blood pressure in the systolic, in the systemic, we have systolic over diastolic, approximately about 120 over 80. This is showing 125 over 75 in this diagram. And it pulsates. And then this is in the pulmonary circuit, much lower pressure system. So how are we doing so far? Kind of review, hopefully. But if you have anything to say, let's chat about that a little bit more. Do it. We're going to move a little bit to the blood vessels now. Some more. We just looked at the overview. So let's look at the types. First of all, the arteries. The very largest of the arteries, and this would include things like the aorta and the carotid arteries going up in the neck to the brain. These are called conducting or elastic arteries. And the reason that we call them elastic arteries is that in their tunica media, they don't have much cardiac muscle, or smooth muscle, excuse me. They have mostly elastic fibers. Say it again, because we kind of messed it up. In the tunica media of the conducting or elastic arteries, they have predominantly elastic fibers, not smooth muscle. They do have a little smooth muscle, mostly elastic. What this means is when the blood gets pumped into these elastic arteries, they stretch out, and because of the elastic, they recoil, and that helps push the blood away from the heart. So these are the very largest of the arteries. When we look at it in lab, histologically, we can stain, and I remember what G word you were trying to say, Van Giesen, right? Probably for elastic?
Could have been.
Maybe, I don't know. We've forgotten already. Anyway, remember you could stain elastic fibers with Van Giesen or semen. That can stain them black or whatever. So we can look at like the aorta and we can stain it and see these elastic fibers if we use an elastic staining. Muscular arteries are also called the distributing arteries. their tunica media, we have smooth muscle. These are most of the named arteries in the body. So if you look at the cadaver and you say, okay, there's the radial artery or the ulnar artery or the musculocutaneous artery or the brachial artery or the femoral artery, any of those are all muscular distributing arteries.
Well, the one I was thinking of is golden.
Oh, okay, that's things, cells, neuronal cells, all black, but it only stains a proportion of them,
Okay.
so that you can actually see them. If it stained them all, it would just be a massive dark.
Yeah, that was the one I was thinking of when I was looking at that in the 5 years.
Oh, it could have been. Yeah. Another G stain. By the way, I should mention, because I sort of forgot, on these blood vessels, most blood vessels have 3 layers. The tunica intima is the one right next to the lumen where the blood is. The tunica media is the middle layer. And the tunica adventitia is the outer layer, connective tissue.
Is the endothelium part of the tunica intima?
That's exactly right. The endothelium is that simple squamous lining cells. It's part of the endothelium. Tunica intima, sorry. The endothelium is part of the tunica intima. By the way, once in a while, fortunately, the tunica intima can also be called the tunica interna. It's the same thing, so don't let that throw you. Tunic means coat, right? And so these are coats or layers. So tunica intima, tunica media, and tunica adventitia. So that's something to think about. Another thing to pick up, since it's here on this slide, the largest arteries actually have blood vessels in their wall, most commonly in their tunica adventitia. And if you have vessels within a blood vessel, it's called a vasovasorum. If you have nerve in a blood vessel, it's called vasa nervosa. So it's interesting that these blood vessels are big enough to need their own smaller blood vessel supply to bring oxygen and things to them. The coronary arteries of the heart would be a type of vasovasorum for the heart. It's the heart's own blood vessels, for instance. And so we see that, and in general that's called a vasovasorum. In the muscular arteries, we've got a lot of smooth muscle in the tunica media, and that helps it constrict or dilate. That helps control where blood moves and the blood pressure. The tunica intima in a muscular artery has a subendothelial, meaning right underneath the endothelium, internal elastic lamina, and that's a good way to help us in one of the ways to differentiate an artery from a vein, for instance. We'll be looking at other ways. Here are some of the differences between the elastic arteries, the muscular arteries, the arterioles, and then a little bit of metarterioles. What is an arteriole versus a muscular artery? It's basically an arteriole is the smallest of the muscular arteries, but we subclassify them as arterioles. Usually, arterioles only have about 1 or 2 smooth muscle layers in their tunica media, where anything over that would be like a muscular artery. We could say the muscular arteries have between about 3 and 40 layers in their tunica media. The very largest arteries, the elastic arteries, have maybe 40 to 50 layers in their tunica media. Obviously, it's going to be easier to see when we're actually comparing these, looking at actual histological images of them. Here's the arterioles. They're not very big, but they do still have layers of smooth muscle. When you go into capillaries only have an endothelium. It has a single cell layer. And that's it. There's no smooth muscle. There's nothing else. So, in other words, the wall gets smaller and smaller as you're going down. Just grab a drink of water real quick. This shows you the aorta. Now we're introducing here, this is from the book, this slide, a new stain that also stains elastic material. But it doesn't stain it black, it stains it purple. This is called the resorcin fusion stain. So we haven't talked about that one before. But you see these purple lines here? These are elastic fibers in the tunica media, and it is counterstained here with this resorcin fusion stain. Here's our tunica intima, you can see with the endothelium, and if you look closely, this little purple line here, you get a little bit of a internal elastic lamina there. And this is your tunica media. You do have some smooth muscle here, but you have quite a lot of elastic fibers. And then you get into the tunica adventitia. By the way, this is a vasovasorum. That's a blood vessel within the wall of a blood vessel, a bigger blood vessel. Now this is a more common, we know this stain better, we're more familiar with it. This is the Van Giesen. Here we abbreviated as EVJ. But this is an elastic stain as well, as you remember. And now this is staining the elastic fibers black. Here we can see very clearly the internal elastic lamina of the tunica intima, and in the tunica media we have the smooth muscle, but we have all these elastic fibers stained black. And then out here we have the tunica adventitia. This is a vasovasorum. This is the tunica adventitia. Notice that this is an artifact that it's pulled apart like this, and like that would all be snug together. This too, but this is a bigger one. This is the tunica media here that we have. This is just a normal H&E prep. This is a muscular artery. So you have your tunica intima, and you have your endothelium, and then here's your internal elastic lamina or membrane, this smooth and lavender structure and tracing. we see that, that's a cue to us that this is an artery and probably a muscular artery rather than a vein. We also have an external elastic lamina between the tunica media and the tunica adventitia, but it is often much less prominent and broken up. And here we see little pieces of it, the external elastic lamina, but it's not this nice continuous ribbon that we're seeing here. This is kind of broken up. These are all nuclei of smooth muscle cells. These are the layers of the muscle. This is a muscular artery. This shows you a muscular artery from your book. The left is H and E, and the right is the same magnification and everything, but it has a Van Giesen staining, so you can see the elastic fibers more clearly. So we see the internal elastic lamina, or membrane, and the external elastic lamina. Notice we don't stain much of the tunica media because we really don't have very many elastic fibers in a muscular artery. These are mostly smooth muscle cells. So that gives you some comparison there. Here you can see, if we look closely, this is another vasovasorum, a blood vessel in the wall of another blood vessel. Here's a muscular artery, and again, this has a Van Giesen stain. And look at the internal elastic lamina. With this stain, it really pops out at us. It's a little harder to see the endothelium because this stain sort of obscures. You can see a little bit of it coming along there. This is the lumen where the blood goes. Arteries like this, muscular arteries, generally have proportionally a thicker tunica media compared to the tunica adventitia. The veins, it'll be the opposite. You have more tunica adventitia compared to the tunica media. Arteries tend to be thicker walled on average than veins, and they tend to have nice rounded contours, but their lumen of arteries is proportionally less in size than the lumen of the veins. In veins, the lumen is big compared to the wall. In arteries, the lumen is proportionally smaller when you compare it to veins, when you're comparing it to the wall. The veins, because the walls are thinner, can tend to be more collapsed or partially collapsed. The arteries tend not to be. They tend to have that integrity. And although you can see occasionally red blood cells and the like in the lumen of arteries, you're more likely to see red blood cells, white blood cells, and the like in veins because the blood is moving slower through veins than arteries. So there are some of the differences, and we'll keep pointing these out as we go through. Here's a muscular artery with just the H and E stain, and here we're seeing the endothelial cells of the tunica intima. Now, if you look here, I'm tracing the internal elastic lamina. That's that kind of reddish structure of this stain along. Then these are your smooth muscle of the tunica media. Here's your external elastic lamina. This one we see it pretty well here. See that? And then here's your tunica adventitia. The tunica adventitia just sort of drifts into surrounding connective tissue. These are fat cells. You probably recognize this is the lumen of the blood vessel. Now we're comparing. This is an artery over here with the A on it, and this is a vein. look at the lumen. If you stretched all that lumen out, it's partially collapsed. But you see the luminal size compared to the wall in the vein? And look at the artery. The lumen is smaller proportional ratio to the wall compared to the vein. And then you have a thicker wall in the artery. And so that shows us some of the difference. We do have an endothelium in both the vessels. And so this is giving us an early view of differences between the arteries and the vein. This is a small vessel here, probably. Veins in the systemic system have valves so that the blood will only go back towards the heart and not away from the heart. Once in a while we'll catch a valve like right here. They look like a little V. The blood can go through one way but not the other way. So this is a valve in a vein. Notice the lumen, very large, and notice the thickness of the wall. We do have some smooth muscle here, not a lot. We have an endothelium and a tunica adventitia. Notice the tunica adventitia is proportionally larger than the tunica media. Again, this helps us understand that it's a vein. Over here we have an artery that has been stained for the elastic fibers. You see the internal elastic lamina is highlighted here. The external elastic lamina is also visible, although more broken up. This is the tunica media with the smooth muscle. Here's the lumen with some red blood cells and white blood cells in the surface. And then a tunica adventitia. This would be a this would be outside. It probably goes to here. There's a little blood vessel here. That's probably a vena, a very small vein. It's called a vena. This is a small artery again with an elastic stain, so we've seen the internal elastic lamina stain. You still doing all right? I'm impressed that you're still awake in the sauna here. Okay. Capillaries are the smallest of the blood vessels. They have only one cell thick, so just an endothelium. There's no tunica media, no tunica adventitia. There's just a lumen and one cell. Of course, they're designed for exchange, right? So that oxygen and nutrients can leave the blood of the capillary and go to the cells. Carbon dioxide and waste products come into the capillary. And so they need to be very thin so that you can have a fast diffusion, efficient diffusion. We have lots of capillaries, and they don't all have an equal permeability. Notice that most capillaries are about between 8 to 10 microns. That's 1 to 10 to the minus 6, if you remember, and about a millimeter long. Some of our sinusoidal capillaries are much thicker in diameter than this, but most of the capillaries are about 8 to 10 microns. Because the red blood cells, do you happen to remember typical diameter of a red blood cell?
This is about 8.
It's about 7, 7.5, so pretty close to that. That means they have to queue up through the capillaries single file. And sometimes they even have to squeeze through. So that shows you how small the capillaries are. And they have to squeeze through like that. The continuous capillaries are, they're all can diffuse through. They all have high permeability. But the continuous capillaries are the least permeable of the capillaries. And then the fenestrated are more permeable. And the sinusoidal are the most permeable. So we're going from continuous, fenestrated to sinusoidal, also called discontinuous to sinusoidal. And they're progressively getting more and more permeable. The continuous ones, you can see where they're found, the nervous tissue, muscle, connective tissue, exocrine gland, the lungs. Fenestrated, endocrine gland, you can see the list here. Sinusoidal are in the liver, spleen, bone marrow, places like that. They just have these endothelial cells. Generally only one red blood cell at a time can go through that small diameter capillaries. Here's the different, three different ones, the continuous, fenestrated, and sinusoidal. Notice that the sinusoidal can be much larger in diameter, maybe about 35 microns instead of about 8 to 10. So these are the large ones, but they're still very thin. The little fenestrations in the fenestrated capillaries, those are little openings, makes them more permeable, but they're only about 70 nanometers, 1 times 10 to the minus 9 openings. So again, continuous are the least permeable, fenestrated, intermediate permeability, sinusoidal, the most permeable. White blood cells can squeeze into the capillaries and then squeeze out by a process called diapedesis. Margination is where they bind, and then diapedesis, they actually move out. A lot of times this is for an infection. They're going to the site of infection and they're squeezing out. After the capillaries, blood vessels form the venules and then the veins. This shows you an electron micrograph of a capillary. This is a red blood cell, and this is a platelet. This is another red blood cell, sort of seen it on the end view. This is a nucleus of the one endothelial cell coming around. There's the mitochondria. You can see that the red blood cell takes up most of the space in the room of the capillary. Again, you can come in here with your arterioles. Little meta-arterioles kind of connect the arterioles to the capillaries, and then it comes through back to the venules. And this shows you the endothelium and the red blood cells and the capillary. This is quite a high magnification. This is 800. This is some little capillaries in the cardiac muscle. That's a good place to find capillaries. So later we'll do the veins, and we'll finish up the veins.